All right, now let's talk about section 2 of chapter 9, the details of cellular respiration, this form of respiration that's going to take advantage of oxygen to extract energy out of foods, out of these energy-rich organic molecules like glucose. So again, here's sort of the overall look, our three steps: glycolysis, Krebs cycle, and electron transport. Um, So think of it this way: we're taking this energy-rich molecule, and through a series of steps, we're going to extract energy out of it. So instead of just extracting it all at one big reaction, we're going to have a series of controlled steps that allow us to extract energy. And it all starts with glycolysis. So here we are, we're out in the cytoplasm of the cell. We have this energy-rich molecule like glucose, for example. And what we're going to do is, now each purple arrow represents, there's about 10 steps altogether in this, but we're just going to distill it down. First, there's what's called the energy investment phase, and that's where a couple ATPs are used to get the process going. You might think of them as something, or a source of energy to sort of get the reaction going, to get over that energy that's needed to get it started. Now, so what happens here is we break it down into two intermediate 3-carbon molecules. So the 6 carbons that were in a single glucose are now into 2 3-carbon molecules. And then we convert those molecules to this stuff called pyruvic acid, or just for short, pyruvate. Okay, those are the same thing. And in that step, we're able to generate our first net gain in energy extracted. So you can see we produce 4 ATP in this step. So we've used 2 up here. So our net gain of ATP is 2 at this point. And we also generate a couple NADHs, a couple of these electron carriers, from this molecule called NAD, nicotinamide adenine dinucleotide. So we take 2 of those and produce 2 of these NADHs, these electron carriers, and as we see here, they are going to go on to the electron transport chain later in the process. Again, keep in mind the overall goal here is to extract energy from the foods we eat and generate ATP, and it's the ATP that is used by our cells as an immediate source of energy to do work. All right, now, but of the energy we started with, most of it is still in these pyruvate molecules. So now we have to break them down to get more of the energy out. All right, so now we're going to move on to the Hans-Krebs cycle. And here we're going to take those pyruvates, and they are now going to be transported into the mitochondrion. And the very inside of the mitochondria, what's known as the mitochondrial matrix, we're going to completely break down this molecule to transfer energy primarily, well, to ATP and more NADH molecules. So you can see there's these initial steps here where the pyruvate comes in, we get our first carbon dioxide being produced. Here we make another NADH molecule. It's going to go off to the electron transport chain. All right, after we've extracted this CO2, we now have this molecule called acetyl-CoA, or acetyl coenzyme A. It's going to enter the citric acid, or the Krebs cycle, which is also known, I should say, as the citric acid cycle, because the first molecule that's made in the cycle is citric acid. And through a series of steps, we're going to produce more CO2. So essentially at this point you can see we've produced 3 CO2s, and this is just showing for one of the 3 carbon pyruvates. Of course there'd be 2 of them for each glucose, but for one of them those carbons that were in the pyruvate now have come out as CO2. And you can see in the process we make a lot of NADH, Again, we make a little bit of ATP here, one for each pyruvate. So for the Krebs cycle, for each glucose that comes in, we're going to again make 2 ATP, one for each pyruvate again. But we're going to make a bunch of these NADH electron carriers and also a different electron carrier here called FADH2. So as a student from a long time ago said, basically in this process, we make a bunch of NADAs and FADAs. That's the goal, these electron carriers. And they're going to take those electrons to the electron transport chain. All right, so we've completed the breakdown of the original molecule. We've produced the CO2s, the waste product, one of the waste products of cellular respiration. We've produced a little bit of ATP here, but a lot of the energy has basically been transferred to these electron carriers. So now in the electron transport chain, this is happening on the inner membrane of the mitochondrium. And when you look at a mitochondria, you've got the outer membrane, and then you've got this rather convoluted inner membrane, which, what are all those folds in that inner membrane, what is the benefit there? Well, lots of surface area over which the reactions of the ETC can happen. So there's a series of molecules embedded on that inner membrane, and all these electron carriers that were produced earlier on converge at the electron transport chain on that inner membrane. Each one of them comes in, gives up an electron. Those electrons are passed along, and just like we saw in photosynthesis, we're going to use the energy of these electrons to actively move these hydrogen ions into the space between the two membranes, the inner membrane space, between the outer mitochondrial membrane and that inner membrane. When those hydrogen ions have been concentrated there, they'll want to move back across the membrane if they can. And again, just like in photosynthesis, they go through ATP synthase to do this. They diffuse back to the inner space of the mitochondrion, the matrix, and in the process of when they move through ATP synthase, we convert ADP into ATP. And because we produce so many of these electron carriers, it's at this step that we can generate a lot of the ATP. The vast majority of the ATP is produced at the electron transport chain. Now, here it is, yes. So this is where the oxygen comes in. Remember our overall formula? We're going to take the C6H12O6 in the presence of oxygen, and we're going to make a bunch of CO2 and water. So here's where the oxygen comes in. Essentially, after the electrons are passed along, oxygen is the final acceptor of the electrons. The electrons start in glucose, they're stripped off into these electron carriers, they're given up to the electron transport chain, and then the electrons end up with the oxygen molecule. Well, in the process of doing that, oxygen not only gets the electrons, but it also grabs onto some hydrogen ions, and so this is where we make our water. So the oxygen is used at the end of the process, and the water is produced at the end of the process. Of course, the glucose, the breakdown of it begins at the very beginning with glycolysis. The CO2 is produced during the Krebs cycle. All right, so here's sort of the totals here. Again, the goal is to make ATP. We make a couple of them in glycolysis, a couple more in Krebs cycle, as well as producing some CO2. 6 of them to be exact. And you can see the vast majority of the ATP are produced with electron transport, as well as using the oxygen to produce some water. Okay, now this is an approximation. it can really range anywhere from 32 to 34, and it depends on how efficient cellular respiration is proceeding. Okay, that's it for section 2.